everyone. This is Jenny Glenn Gingery from the Endocrine Society. We're going to be starting in just a minute here. Looks like most of the people who registered are online already, so thank you for being prompt. Just a couple of housekeeping announcements before our presenters, Dr. Patisaul and Dr. Vandenberg, get started. Basically, we've got everybody muted except for our panelists. We've done that just to eliminate any distracting noises like typing, dogs barking, sirens. If you do have any questions, feel free to type in the chat window. If you're having any technical issues, you can email us at media@endocrine.org, and we'll try to get that sorted out for you as quickly as we can. We will have both of our speakers present. They'll be doing a little bit of back and forth during the presentation, and then we'll be taking any of your questions at the end. This webinar is being recorded, so you'll be able to visit our website to hear any points you may have missed. We'll be reading the questions out loud for the benefit of journalists who may be listening to this recording after the fact. So I'd like to welcome our two speakers. Laura Vandenberg has analyzed the results from the FDA's core report from the Clarity BPA study, and we're very pleased to have her. And then we also have Clarity BPA investigator Heather Patisaul. Both Dr. Vandenberg and Dr. Patisaul are members of the Endocrine Society's Endocrine-disrupting chemicals advisory group, and we're very pleased to have you both here today. So first, I'd like to go ahead and turn things over to Dr. Vandenberg.
Great, thank you, Jenny. As Jenny mentioned, we're going to go back and forth between myself and Dr. Patisaul a couple times, just because we thought this was the easiest way to present the information to you. So starting out, I think it's important to sort of talk about what do we mean when we're talking about safety and also toxicity. So the reason why we decided to organize this webinar is that the FDA has repeatedly stated that the way that BPA is used in food containers is safe for humans. The problem with that statement is that it's based on incomplete data. And the Clarity study, which we'll talk to you about today, has been ongoing since 2012 in one form or another. And it involves data that was collected both by the FDA and by academic scientists. While the FDA's data are available, the whole point of the study was to include also academic data, and some of that is still undergoing peer review. So everything that we're going to talk about today will focus on what's available publicly, but you should know that there's also additional data that is still forthcoming. Even considering the fact that the data that have been presented by FDA have been interpreted by FDA as suggesting that BPA is safe, scientists still disagree, including myself and I believe Dr. Patisaul, as you'll hear from us. And so what we want to talk about is, why does this science suggest that there is cause for concern? Next slide, please. So what is BPA? BPA is a compound that is widely used in consumer products. The concern that we have about BPA is that for more than 30 years, it's been known that BPA mimics estrogen when it's found in the body. And it does this by binding to the estrogen receptor. It also binds to other hormone receptors in the body and has other effects that are unique from the effects of estrogens. BPA has been linked to a wide variety of effects. Associations have been shown in human epidemiology studies, more than 100 human epidemiology studies, suggesting that people who have more BPA in their bodies are more likely to have certain reproductive disorders like polycystic ovarian syndrome, that children with higher levels of BPA exposure are more likely to have behavioral problems, and that both children and adults that have higher exposures to BPA are more likely to have metabolic disorders like obesity and abnormal liver function. Next slide, please. We're exposed to BPA because it's used in all of these consumer products, including metal cans that are used in food packaging, a lot of plastics, including plastic water bottles, not usually the ones that are used once and then thrown away, but more the sports kind of water bottles, a lot of toys, medical equipment, and even thermal receipts. We know that BPA leaches or comes off of these products when they're used even according to manufacturer's instructions. So for example, metal cans have a lining in the insides of them that prevent the food from interacting with the metal and preventing that sort of tinny taste that that food used to have before BPA started to be used as a resin. BPA is not actually the ingredient that's sprayed on the inside of those cans, but it breaks down into BPA, or if it's incompletely polymerized, it will be released as BPA into the food. And the CDC has a very large biomonitoring study that has been done that's looked at the general American population and has found that BPA is found in the bodies of more than 95% of people at any one time. Of course, some of us might not have BPA in our urine right now, but that doesn't mean that we didn't have BPA in our urine yesterday, that we won't have BPA in our urine tomorrow. Next slide, please. So as I started to say earlier, there are hundreds of epidemiological studies that have now been done that have tied BPA to specific health problems. In 2014, a review conducted by authors at the Endocrine Disruption Exchange evaluated 100 epidemiological studies that had been published at that time that suggest associations between BPA exposure and those range of outcomes that include neurological outcomes, abnormal metabolism, reproductive effects, as well as growth and development effects. Next slide, please. So where is this disagreement coming from? I think I'm supposed to also pass this slide off to Dr. Patisaul to talk about, but a major part of the problem has been not what is available in the scientific literature, but who is reading it, how are they interpreting it, and how are they using it for decision making. Dr. Patisaul?
Thank you, Laura. So this slide shows the first sort of page of a long document that was produced by the Food and Drug Administration, which illustrates this issue of how you can get disagreement about data. And so this was the latest scientific review they did. So they did a literature search on bisphenol A to make an evaluation about its use, the safety of its use in food contact materials. And if you read through this very long document, you'll find that they found over 300 studies in the literature that they pulled for this review. But as they go through study by study, they describe the strengths and weaknesses of every study. And of course, every study has strengths and weaknesses. And then ultimately evaluates whether or not they can be used for risk assessment. And for example, within the neurotoxicity section, they identified 36 studies, and none were identified as being useful for risk assessment, except for one, which was an industry-funded study and was an acute high-dose exposure to ovariectomized female rats. So one would argue that that's not quite the right animal model to address neural toxicity, and that if you're only looking at 1 out of 36 studies, it's going to give you a very different perspective than if you had looked at all 36. Similarly, for the reproductive endpoints, they identified 25 studies and identified none of them for risk assessment. And that's true for 13 other endpoints. So the lens that we use to evaluate the studies that are available in the literature can certainly frame and shape our opinion. So that's represented very nicely in this pie chart. If you take only a sliver of the pie to make a decision about what kind of pie that is or what it looks like, you might get a very distorted viewpoint than if you looked at a bigger piece of that pie. And for some pies, like the reproductive effects and other effects, they actually took no data whatsoever from the literature to make their evaluation. Next slide. So this frustrates scientists like me who try to generate data to be used for these types of evaluations. And so here are two. One was done by the National Toxicology Program, which is located within NIEHS here in Research Triangle Park, North Carolina. And the other one was an expert panel that was convened by the World Health Organization. Both of these, so these are not regulatory agencies, they're scientific bodies, but they do not make regulatory decisions about chemicals, also had trouble combining the literature. They used different approaches. They took a wider viewpoint of that literature and came to different conclusions. So these two documents both concluded that there was some concern about bisphenol A on brain and behavior, but that they identified that there was a big problem with trying to pull all this literature together because the experimental protocols were different, the animals were different, the dosing was different. It was not a very harmonious literature. And so Clarity BPA was designed to address this problem. Next slide. So Clarity BPA is the most ambitious project that's ever been done to look at the health effects of pretty much any chemical. And so it was designed by consensus. And so it was designed to allow scientists like me generate data that could be used in risk assessment by FDA. And so that study design is outlined in this report, which is available from the National Toxicology Program. They are coordinating all aspects of this study, and it involves scientists from academic institutions, multiple academic institutions, as well as the NTP and the FDA. And so the goal was to create this culture of partnership and communication between the agencies that have to make these decisions about safety and the scientists who are producing the data that's trying to inform those assessments. And so this just kind of diagrams the alphabet soup that is Clarity BPA. So NIEHS is a division of the National Institutes of Health that is the only branch that is not in Bethesda, Maryland. It is here in Research Triangle Park, North Carolina. And the National Toxicology Program is located inside that, inside NIEHS. And then NIEHS gave grants, like NIH gives grants to many investigators. NIEHS gave grants to 12 extramural grantees. And so I was one of those grantees. We had to submit our grant. It went through peer review. It was also evaluated by FDA and NTP for feasibility. And so we were selected by NIEHS to be funded for this project, which was the academic piece of Clarity BPA. And then the Food and Drug Administration has its own research institute, and that's NCTR, or the National Center for Toxicological Research, which is located in Jefferson, Arkansas. And so those groups all came together to produce animals and data shared between everyone, which is the Clarity BPA program. And so I'm going to hand it back to Laura, who's going to go over the basic study design and talk about some of the results that came from the core study, which is the piece of the study that was done at NCTR in Arkansas. Dr. Vandenberg?
Great, thanks, Heather. So Clarity BPA, as Heather just mentioned, is one of the largest, if not the largest, research undertaken by this multi-agency collaborative group. And part of the reason why it was such a large study is shown here on this slide. This illustrates the fact that within Clarity BPA, there were really a handful of experiments that were going on. In one that's labeled the continuous exposure, animals were fed BPA starting at day 6 of pregnancy, and then they gave birth and their offspring began to be exposed, again, fed BPA daily from birth until either they were euthanized at 1 year or 2 years of age. And when I say fed, what I really mean is force-fed. This is a protocol that's called gavage, where BPA was inserted directly into the stomach of the animal through a little tube placed in their mouth. In addition to animals that were continuously exposed to BPA throughout their gestation and entire life, there was also a second group of animals that were exposed only during early development. So again, the mother was force-fed BPA starting at day 6 of pregnancy. When she gave birth, the pup started to be force-fed BPA until they reached day 21 of life. This is prior to puberty. After that point, they were no longer exposed to BPA. There were 5 different BPA dose groups, as well as an unexposed control. And there were also animals that were exposed to ethanol estradiol, here indicated by the letters EE. They were exposed to one of 2 doses. The EE animals were only exposed continuously, so there were no stop dose EE animals. So this is a really complicated study design. And the animals that were produced from this experiment had to be allocated both to the FDA's endpoints that they were going to look at, as well as to the academic researchers. The FDA's endpoints were all collected at either 1 year of age or 2 years of age. But as Dr. Patisaul will explain to you at the end of this talk, the academics asked for animals at all different life stages, depending on what kind of outcomes it was that they were looking at. Next slide, please. So the scope in general of the Clarity BPA study is that there were outcomes that were evaluated in the core study, the study conducted and analyzed by the FDA. Those are indicated here with the term guideline. Those were normally outcomes that were related to overt signs of toxicity. Things like massive changes to the weights of organs, very overt signs of abnormal behavior like seizures, changes to the body weight of the animal, changes to the number of pups that were born to exposed females. There were also, of course, the academic outcomes, which were designed based on previous analyses that had been done by the academics, previously published literature, as well as very specific hypotheses. Next slide, please. So looking very broadly at the core study, the outcomes that were evaluated by the FDA. And again, these are normally overt signs of toxicity, things like changes to the weight of organs, tumors that were forming, changes to massive inflammation in different organs, and shown across the top are the different doses from low to high for exposure to BPA. And what you can see is that in the core study, the FDA study, there were significant effects on numerous organs. And of course, within a certain organ, like the reproductive tract, for example, there are multiple things that were evaluated within the reproductive tract. So we've collapsed here a lot of different effects into a single data point and asked, were there significant effects at that dose or not? Wherever you see a cell in this table with a color, either red for females or blue for males, is where there was a significant effect detected in the FDA's study. Next slide, please. So I just want to give a very brief overview of some of the effects of BPA that were observed in the FDA's study that I thought were very serious. And what was perhaps surprising to the FDA was that these outcomes were typically seen in the lowest doses that were examined. Next slide, please. So one example is the incidence of mammary cancer. This was really only seen significantly increased at the lowest dose of BPA that was observed, and only in the animals that were exposed during development. There was also significant increase in inflammation of the lobes of the prostate, again seen at the lowest dose group in animals that were exposed throughout life. Next slide, please. There were significant increases in kidney nephropathy in females only, and in the females that had been exposed to the lowest dose group continuously throughout life. Nephropathy, for those of you that are not in the medical community, the nephron is the functional unit of the kidney. So this is a damage or destruction of the functional part of the kidney. There was also an increase in body weight in females. It was seen really in females in older age if they had been exposed to 250 micrograms, one of the lower or mid-lower dose ranges, if they had been continuously exposed. Next slide, please. So this is a graph that's illustrating the incidence of mammary cancer in animals that were exposed to BPA. And this is only looking at animals that were exposed during early development. And what you can see is that more than 20% of animals exposed to the lowest dose of BPA and more than 15% of animals exposed to 2,500 micrograms per kilogram per day BPA had increases in the incidence of mammary cancer compared to controls. Next slide, please. A similar low-dose effect seen at the 2.5 microgram per kilogram dose group was seen in the incidence of prostatic inflammation. So this is considered a risk factor for prostate cancer. And in fact, more studies that are forthcoming and that I know are currently in press are suggesting additional effects on the prostate. Next slide, please. So why is it that these effects were seen not just in the Clarity BPA study, but specifically in outcomes that the FDA evaluated? And yet the FDA seems to dismiss low-dose effects. And we know that they're dismissing those low-dose effects because they're suggesting that, that people are fine using food containers that release BPA and that expose people in the range of those low levels. Well, I think that one of the reasons why FDA is dismissing those low-dose effects is that there's an expectation that with increasing dose, there should be an increase in an effect. So, for example, the more cake that you eat, the fatter you should get, or the more poison that you drink, the sicker you should get. This is a general expectation when you're talking about something that is acting strictly as a toxin. So the more that you are exposed to that compound, the sicker you should feel. Next slide, please. In actuality, with hormones, endocrine disruptors, as well as vitamins, other essential nutrients, a lot of drugs and pharmacological agents, the reality is not a linear relationship between dose and effect, but instead what often looks like a U or an inverted U-shaped relationship. And the reason why is that a chemical like BPA in the low dose range is acting like a hormone, usually like an estrogen. At the high dose range, BPA could be acting via other hormone receptors, via other mechanisms, or it could just be acting as a toxicant. So several people have asked me, why do I think that BPA could induce mammary tumors at the low doses, but not at the high doses? If the cells that are being targeted by BPA are proliferating in the low doses, but are being killed off or otherwise suppressed at the higher doses, we would see a relationship like this. And in reality, again, this non-monotonic, this inverted U-shaped response has been reported for estrogenic chemicals in the mammary gland, and in fact is well known for breast cancer patients that are treated with drugs like tamoxifen. Next slide, please. So just some broad conclusions from the Clarity FDA core study. Number one, I think that there are serious effects that were observed in the FDA core study, even in the groups that were exposed only to low doses of BPA. Unfortunately, these effects seem to be dismissed by the FDA, often because they aren't observed at high doses, and the FDA takes a traditional approach where they expect that toxic effects should be getting worse at higher doses. That just isn't true for chemicals like endocrine disruptors, such as BPA. Next slide. Another thing that I haven't discussed at length here, but I think is very important, is that the Clarity study included ethinyl estradiol as a positive control. And the reason why I think that's important is that prior studies have been done on ethinyl estradiol. And so now we have the Clarity study that included ethinyl estradiol and prior studies that looked at similar outcomes that also evaluated ethinyl estradiol. And what I was shocked to find is that these studies were not replicable. The outcomes were not showing the same effects of ethinyl estradiol in different studies, even though those studies were conducted in the same NCTR laboratory with the same rats. So that to me suggests that there's a much deeper story here, that the way that regulatory agencies have been evaluating chemicals for safety using these traditional methods of evaluating toxicity may not be reproducible. Next slide. So I'm going to pass it back to Dr. Patisaul, who will tell us about the academic outcomes or academic studies that were included within Clarity.
Yes, so to build on that theme of reproducibility, there's been multiple studies in Clarity now that have been published, and I'm not going to go through them all. Instead, I'm going to talk about this graph, which was put together by Carol Kwiatkowski at the Endocrine Disruptor Exchange. And what it shows is the availability of the literature and which studies, the proportion of studies that show effects and the proportion of studies that do not show effects. And so what you can readily see is that in most cases, brain and behavior and the female reproductive system seem to be heavily impacted by BPA. And that's exactly what the Clarity academic studies have found. So I have published 3 studies in Clarity on the brain now. All 3 of them found effects of BPA in the brain. Other laboratories have published effects on the female reproductive system, including the ovary, and all of those have found effects of BPA. If you look at the yellow boxes, the organ and system outcomes, and focus on what some of those organs are, by and large, you're looking at the heart. And the final BPA study that is now published and available has again shown effects that there are BPA-related effects on the heart. In all cases, those effects are at low doses. So when you think about reproducibility in the broadest sense, and you look at the effects that the FDA found at low dose, you look at the effects that the Clarity investigators found at low dose, and you go back and look at the existing literature, you see a very clear picture of BPA-produced effects on brain and behavior, female reproductive system, and the cardiovascular system. Next slide. And so here's the timeline of where we are and what's to come. So we're at the integration phase for BPA Clarity. FDA has finished and published its report. Some of the Clarity investigators have finished. Some of those studies are published. Some of them are forthcoming. But all of the data is now available through NTP. So all of the investigators agreed to make their raw data available, and it is now freely available online. And so that's the little bullet that says grantee data rollout begins. And now we're in the very critical phase where we have to take all of that information, put all of that data together, and come up with a conclusion for how BPA impacts our bodies. And so that will involve not only the FDA's core study data, but all of the data from the independent investigators. And so that's why there has been concern from the Endocrine Society and others that FDA is jumping the gun a little bit in its conclusion that BPA in food packaging is safe, because the data has not yet been integrated. And so National Toxicology Program will be in charge of integrating this data set. They are still finalizing the methodology for how they're going to do that, but that final report should come out in the fall of 2019. Next slide. So what is at stake? Well, a lot. It's very expensive to have sick people. Whether you're in the EU or in the United States or anywhere else in the world, we know that chronic illness is expensive. And so I'm going to hand this back off to Dr. Vandenberg, but the take-home point is that the cost of doing nothing about these chemicals is significant, and it's costing us quite a lot, not just in terms of our health, but also in terms in real dollars for healthcare costs. Dr. Vandenberg?
Yeah, we wanted to just finish up this talk by referring you all back to previous work that was published in Endocrine Society journals, led by our colleague Leo Trisande. And all of this is not focused on BPA per se. This is really looking at several endocrine disrupting chemicals. On the right-hand side of this graph is showing you the kinds of endocrine disrupting chemicals that Leo and his colleagues considered in their evaluation, including pesticides, flame retardants, and other chemicals that come from plastics like phthalates. And what they concluded was that the costs of exposures to endocrine disruptors are actually huge. In the EU, somewhere between 150 and 270 billion euros every year. In the US, the effects are driven by somewhat different chemicals and somewhat different health outcomes, because there are US versus EU differences in which chemicals are used and also in our rates of diseases. But the numerical outcome is very similar. We often hear about what would the cost be to ban a chemical? How would that affect the industry? And in fact, a classical example of this was the discussion that rotated around whether we should ban asbestos and how much that would cost the asbestos industry if it was regulated. What we don't often talk about is what is the cost of inaction? What is the cost of deciding to keep a chemical on the market? And Leo Trisande and his colleagues really are trying to get at that answer. And the answer is that continuing to use chemicals like BPA that even though there is not a 100% causal relationship between BPA and specific health outcomes in humans established, that the best data that's available suggests that exposures to endocrine disruptors like BPA are costing us billions of dollars a year. And so I think it's just important to end on the idea that the decision to do nothing is a decision, and that decision does cost us something. So I think we will end there, and we're happy to take questions. And we'll turn this back to Jenny, who's going to run the Q&A session, hopefully with some order.
Thank you so much, Dr. Vandenberg and Dr. Patisaul. We really appreciate you taking the time today. I just want to go ahead and open the floor for questions. Just to keep everything in orderly fashion, we're just asking if you could please type your question into the chat window, and we'll go ahead and read it out from there.
Sure, and this is Colleen. While we're waiting, I can just ask a couple of questions that we had on our end while everyone's typing in their questions. So to get us started, the first question we have here is, what do you hope policymakers will learn from the Clarity BPA study?
This is Dr. Patisaul. As someone who participated in this study, I think we've already learned a lot. Cooperation is going to be a big one, and that's got to be both for the regulators and for the scientists like me who had to learn how to generate data that regulators could use. I think they also need to pay attention to which endpoints are sensitive and get real about updating the toxicity testing strategies that we have in the regulatory arena. If the goal is public health, and I think most scientists genuinely want to improve public health, then we have to improve the testing. And a significant advantage of the Clarity BPA program is we really got an opportunity to kind of test drive some of the endpoints and assays we think are going to be the most sensitive. And so if there was one takeaway that I would really like regulators to have, it would be that they need to use that information to update their testing systems.
I'll just reiterate that point, and also that I think part of what is now leading to some of the frustration is that that were agencies involved in the testing of BPA that are also involved in its regulation. And that may be something that we want to avoid in the future, that it may be best to bring in groups like the NTP to run the animal studies, because the NTP doesn't regulate any chemicals and therefore has less of a vested interest in maintaining the status quo.
Okay, great. We actually just got our second question in from Dennis Thompson at Health Day. And his question is, why do you think the EPA jumped the gun? Also, what can consumers do to avoid BPA? And is info on BPA typically included on product labeling if a person wants to avoid it?
So this is Dr. Patisaul again. I think that the jumping of the gun by FDA, it was one person at a pretty high level who put out a press release. I don't know that that opinion is universally held by all members of FDA, but regardless, I share Dr. Vandenberg's concern that FDA has put a stake in the ground. They've said that BPA is safe, and they're very reluctant to back away from that stake, because doing so requires them to essentially capitulate that they got it wrong the first time. But science should always be movable. We're always discovering new data. And so for many of us that are scientists, the idea of changing our minds is just natural. We do it all the time. Maybe there wasn't enough data the first time to come to a conclusion, but there certainly is now. In terms of how to prevent exposure, it's pretty easy. BPA dissipates in the body really fast. So if you use less plastic, shift to more glass, try not to use thermal paper receipts, don't put your plastic containers in the microwave, you actually have pretty good control over, reasonable control over your exposure to BPA. It's difficult to know which of the can linings in canned foods have it. Some cans that say BPA free actually have a chemical that looks almost exactly like it as a substitute. So if you're really worried, then you can avoid canned foods, particularly things like tomatoes that are acidic and react with the lining. And that's a good way to reduce exposure. Dr. Vandenberg?
Yeah, I think the question about product labeling is really important because chemicals like BPA do not have to be labeled on products. What we instead see is a lot of labels that will suggest that a product is BPA free. So we're often not aware of where our exposures come from. And as Heather just mentioned, when something says BPA free, what our concern is that a lot of the replacement chemicals that are being used to substitute for BPA are just as bad, if not worse, and we often know very little about them. These are often sort of referred to by scientists as regrettable replacements. We just don't know that we're regretting them until enough time passes so that we can study them. And of course, with the thousands of studies that are now available on BPA, and yet we haven't done anything about them, the concern would be that we're shifting from BPA not to one replacement, but to sometimes 20 to 50 different kinds of replacement chemicals. In 20 years, think of how much work has been done on BPA. How long will it take for us to study well all of those replacements? It's sort of mind-boggling to think about.
So we have our next question from Kristen Jenkins at Medscape Medical News. And her question, it's actually a three-part question. What can clinicians do? What should they be telling their patients? And what are the implications for all FDA drug, chemical, and toxicant testing?
Wow, that's a huge and super important question. It is. I mean, so I'm not a clinician. I'll just start with that. But I think generating awareness, I think it's wonderful for clinicians to have a dialogue with their patients, not just about bisphenol A, but chemicals in general. I mean, it's really sad to say, but just because something is on a store shelf doesn't mean that it's safe. So people have to become a little bit more proactive when they go shopping. And there are tons of internet tools and other tools available to do that very smartly. Environmental Working Group has a wonderful app called Healthy Living that helps people navigate the grocery store. It'll help direct you to safer cosmetics, safer personal care products, safer cleaning products. I personally use it. I know other clinicians who have recommended it to patients, but just having that dialogue and being aware, pesticide use in the home, and thinking about environment as a whole as it contributes to health, I think is really smart. And then the question about regulatory testing in general is the sort of big elephant in the room. And, you know, the drug side of FDA is very different. I don't want to speak to that, but at least on the toxicity testing side, I mean, again, I think we have to get very real about trying to update the methodology by which we do this. We're certainly capable of narrowing in and identifying potential toxicants. The problem is getting that data through risk assessment, either because it's deemed unacceptable for risk assessment, like in the study that I showed you, or because the type of testing that's done at regulatory agencies is not very good and it's old and outdated. So we need to modernize that system. We're in a desperate need to modernize that system. And I think we're very capable of doing that. And Clarity BPA shows us how. Dr. Vandenberg?
Yeah, I would say that the Endocrine Society is developing tools. So there's a task force that's currently put together to develop tools to help clinicians to just think about endocrine disrupting chemicals, as well as ways that they can talk to their patients about them. We are absolutely aware that most physicians get 15 minutes with their patients every year, and they have to talk about everything from drug and alcohol use to seatbelts to domestic violence, which then sometimes makes it seem like, where am I going to fit in a conversation about BPA? And I think that's a very realistic, honest issue that we have to be aware of. That's challenging for clinicians. What I do know is that there have been some tools developed to help clinicians to take environmental histories from their patients, and whether that should be done for all patients or for patients who have very specific concerns is something that needs to be sort of discussed and evaluated with data, evidence-based decision making. But there are tools that are available, and I know that other academic and medical societies like the American College of Obstetrics and Gynecologists is also or has already developed materials that clinicians can use. So it's a matter of really educating the clinical groups for what's reasonable to address in a 15 or 20-minute yearly visit.
Okay, great. And we have a couple of questions here as well from Benedette Kafari at newsmedical.net. Her first question is, how do your BPA doses compare to human exposure doses? And the next part of the question is, do you have any information on what type of scientific methods are used to measure the amount of BPA that leaks from certain food packaging materials? And does the FDA have any recommended techniques that they use prior to approving the use of these products? I'm happy to repeat that as well, since I know that was a longer one. So the first part of that again was, how do your BPA doses compare to human exposure doses?
Okay, so that's a good question. Go ahead, Laura. Do you want to take that one?
Well, I was just going to say that really dose is such a complicated thing because we can compare intake, right? Like, what do people intake versus what were animals sort of force fed, but that doesn't take into the account the fact that rodents generally metabolize chemicals faster. So if you're talking just about intake, the lowest dose is the one that's actually relevant to human exposures, or most relevant to human exposures. 2.5 micrograms per kilogram, the lowest dose in Clarity, is right around what people think that children are exposed to. It might be just a tad bit higher, but right around there. But it probably takes doses in the next 2 dose ranges to replicate in the bodies of creatures the levels that are actually seen in people, just because rodents do metabolize the chemical faster. So the next question was about methods for testing leaching from products. That is not something that's typically done by regulatory agencies. It's done by scientists. And so there are actually several dozen papers that have looked at BPA leaching from food contact materials, thermal paper, medical equipment. They usually use analytical chemistry methods, so they'll do some form of extraction, and then usually HPLC or liquid chromatography, and then usually tandem mass spectroscopy. So, very precise and accurate methods, usually that can measure as low as 0.1 part per billion. So they can really detect what's in food or what's leaching out of cans. To my knowledge, the FDA does not routinely test foods for BPA, because BPA is an approved use for food contact materials, and they're not tested. So it's hard to know how to compare one can of green beans to another can of green beans unless you have usually an academic scientist who's going to measure and compare them. I don't know if you have anything to add there, Dr. Patisaul.
Oh, that was perfect.
I think that's all the questions we've received. Thank you again to everyone who joined us for this discussion, and thank you to Drs. Vandenberg and Patisaul. We really appreciate you taking the time to educate us all about this topic. So again, thank you to everyone who joined us for this webinar. We will be posting it on our website for anyone who wants to go back and review any of the materials. And really appreciate everyone attending. Have a great day.
Thanks, Jenny.
Thank you.